I am Dr. Sandeep Kumar Mishra, and I am here to discuss about the various aspects of the pharmaceutical microbiology, how microbes are important in the field of pharmaceutical engineering. So, one of the requirements to study the bacteria is to identify them. So, how do you identify bacteria or different types of microbes? So, to identify or visualize the bacteria, we use one term called stain. So stain is nothing but it is a substance that adheres to the cell and gives color to the bacteria. So we can identify the bacteria on the basis of the color which is imparted by the stain to the bacteria, and then we can study the different types of bacteria on the white or the transparent background. So simple staining involves staining of the microorganisms with only single type of stain. So in this type of staining, only the microbes are stained with single stain, and usually methylene blue or basic fuchsin are the stains which are used for staining the microorganisms. And then the second type of the stain is the differential stain. So differential stain are those types of stains which can be used to identify different types of microorganisms or to differentiate between the different types of microorganisms. So if we discuss about the differential stain, one of the most common method of the differential staining is the Grams staining. So Grams staining is identified by Christian Gram, and it is used to identify or differentiate between the Gram positive or the Gram negative bacteria. So Gram positive bacteria are those bacteria which are having peptidoglycans in their cell wall, whereas Gram negative bacteria are those bacteria which lacks peptidoglycans in their cell wall, but they have ample amount of lipopolysaccharide layer in the plasma membrane. So if we see how the Gram staining works, then initially the mixture of the bacteria is fixed, and then those fixed bacteria are then stained with the primary stain which is called the crystal violet. So when we stain the bacteria with the crystal violet, all the bacteria gets colored with violet colored with the crystal violet stain. Then we use a suitable fixating agent. Here the fixating agent used is iodine. So iodine specifically fixes the crystal violet stain to the cell wall of the different types of bacteria. And then finally we use decolorization agent. So decolorizing agents may be acetic acid or ethanol. Using decolorizing agents, the Gram negative bacteria are unable to hold the crystal violet stain. So with the decolorization, the decolorizing agent stains away the crystal violet which is present in the cell wall from the Gram negative bacteria. However, the crystal violet remains stained or remains present in the Gram positive bacteria. So all the Gram positive bacteria gets stained with the crystal violet, whereas after decolorization, all the Gram negative bacteria loses their stain. So to stain the Gram negative bacteria, then we use a counter stain, which in case of Gram staining is safranin. So safranin then stains the Gram negative bacteria with the pink color. So in this way, Grams staining is used to differentiate between the Gram positive bacteria and the Gram negative bacteria. Gram positive bacteria gets stained with the crystal violet stain, hence appear blue in color, whereas Gram negative bacteria gets stained with the counter stain, hence they appear in pink in color. So another stain which is used to identify microorganisms, specifically Mycobacterium tuberculosis, is the Ziehl-Neelsen stain. So Ziehl-Neelsen stain is used to differentiate the Mycobacterium species from the other bacteria. So in this case, the primary stain which is used is the carbol fuchsin, whereas the counter stain or the secondary stain which is used is either the methylene blue or the malachite green. So we can see that we can easily identify bacteria through the staining, but microorganisms are not always beneficial for the human body. Some microorganisms are beneficial, whereas some microorganisms are pathogenic in nature. Hence, their entrance into the human body needs to be protected. So how we protect the entrance of the microorganisms into the human body? Either we should take some proper care, or we should make a condition where these microorganisms cannot enter into the human body, or they cannot penetrate to the body through various other means. So one of the best methods to remove the microorganisms is to completely degrade or destroy the microorganisms as well as spores. So this process of complete degradation of the microorganisms or complete removal of microorganisms from the surfaces or even the spores which are responsible for growth of the bacteria is called as the sterilization. So sterilization is very important as it prevents the contamination in sterile products. Also, sterilization helps in the prevents the transmission of pathogenic organisms, microorganisms, which are responsible for causing disease in plants and animals. So how microorganisms are killed during sterilization? So during sterilization, either there is denaturation of the bacterial proteins, or there is interference with the protein synthesis, or interference or interruption of the DNA synthesis, oxidative damage of the cell, or disruption of the cell membranes. So there are various factors which affect the sterilization. These factors may be the contact time, the physicochemical environment, presence of organic material, or the temperature, the type of microorganisms, the number of microorganisms, and the material composition. So if we talk about the different methods of sterilization, the sterilization methods can broadly be divided into physical methods. So the physical methods involves dry heat sterilization, or it may involve wet sterilization, which involves autoclaving. Then there may be sterilization by radiation, like ultraviolet rays, or ionizing radiations like X-rays or gamma rays can be used for the sterilization. And then one other method for the physical method for the sterilization is the filtration or mechanical method. So this method involves the filtration with very high quality membrane filters, which selectively prevents the transfer of the bacteria through its pore. And then second is the chemical method. So chemical method involves the use of various chemicals like ethylene oxide or formaldehyde, or there may be sterilization by disinfectants like alcohols or phenols. So if we talk about one of the major organisms which is the link between the non-living and the living thing is the virus. So virus is a link between the living thing and the non-living thing. If we talk about the structure of the virus usually have the outer surface of the envelope protein or viral genome. It may be DNA or RNA. Then there is membrane covering the viral DNA, which is called as the nucleocapsid, and then there is viral tegument. So if we discuss about the reproduction of virus, there are different phases. First is the attachment, then penetration and release of the viral DNA, and then step 3 involves the reproduction of the new bacteriophage or the new virus inside the bacterial or the host cell, then assembly of the various viral components, and then finally the release of the newly formed virus out of the body. So disinfectant, if we talk about the disinfectant is the process of destruction or removal of microorganisms and reducing them to the level not harmful for the health. So if we talk about the difference between the sterilization and disinfectant, sterilization is just complete removal of microorganisms, whereas disinfectant is just reducing the number to the level so that it may not be the pathogenic in nature. So what are the various factors affecting disinfections? Again, these are similar to what are the factors affecting sterilization, like temperature, time of contact, pH, surface tension, formulation, or the chemical structure of the disinfectant, types and number of microorganisms, interfering substances, and then potentiation. So if we talk about how we identify whether what is the strength or what is the potency of the disinfectant, then various methods are used, but one of the two most important methods used are the tube dilution or agar plate method, cup plate method, or the ditch method. So in the tube dilution method, the bacterial are grown in the various different types of tube, and then various concentration of the disinfectants are added into the culture tube having bacterial growth, and then we see for the inhibition of the bacterial growth. So the concentration of the disinfectant, which is the responsible for inhibiting the growth, is determined on the basis of the minimum inhibitory concentration. Then another method is the cup plate or the cylindrical method, which is used for the evaluation of the disinfectant action. Here the bacterial colony is grown in the petri dish, and then cups are bored, and in this disinfectant is kept, and then the zone of inhibition appears and we measure the length of the zone of inhibition. And then finally, one of the most important method is the ditch plate method.